In 2026, we have discarded the archaic myth of memory as a static filing cabinet. Storage is no longer viewed as a passive act of putting data into folders. Instead, memory is a dynamic, reconstructive performance. It is a biological event of refiring neural circuits, where every act of remembering is effectively a creative reconstruction. Synaptic plasticity is the foundation of learning. Long-term potentiation, or LTP, is the process where synaptic connections are physically strengthened through repeated activity. When a presynaptic neuron is stimulated repeatedly, it releases a surge of neurotransmitters into the synaptic cleft. This activity triggers intracellular pathways that insert more receptors into the postsynaptic membrane. This increases the sensitivity of the receiving neuron. The result is a stronger, more efficient connection. The synapse has been potentiated, allowing the memory trace to persist. The engram is the physical trace of a memory. Rather than storing information in a single location, the brain creates an ensemble of neurons, known as engram cells. When a unique experience occurs, these specific neurons strengthen their connections, forming an ensemble. This pattern represents the memory. These cells fire together, acting as a functional unit. By reactivating this specific circuit, the brain reconstructs the memory of the original experience. Engrams serve as the biological foundation for our past, linking neural activity to the persistence of individual identity. The hippocampus serves as the brain's primary fast learning mechanism. It functions like a temporary scratch pad, capturing complex experiences almost instantaneously. When sensory input arrives, it is processed rapidly by the hippocampus. These data points are encoded as distinct neural patterns. However, this workspace is physically limited in capacity. Think of it as a temporary buffer. Information is recorded quickly to prevent loss, but it must be transferred elsewhere for long-term storage, as the hippocampus cannot hold infinite data. Systems consolidation is the architectural process by which the brain migrates information from the hippocampus to the neocortex. The hippocampus serves as a fast learner, capturing experiences quickly. However, this storage is temporary and fragile. During sleep, the brain initiates sharp wave ripples, high-frequency bursts of activity that replay neural patterns. This data transfer allows information to be integrated into the broad, stable networks of the neocortex, ensuring long-term memory stability. Ultimately, systems consolidation is what transforms a transient daily experience into a permanent part of your knowledge base. The brain is bombarded with sensory input every second. If all synaptic connections were permanently strengthened, the system would be overwhelmed by noise. The synaptic tagging and capture hypothesis explains how the brain selectively solidifies only important memories. When a specific experience occurs, it activates distinct neuronal circuits. These activated synapses undergo a chemical marking process. We call this a synaptic tag, an amber marker that identifies these sites as needing stabilization. Simultaneously, the cell body produces plasticity-related proteins. These proteins circulate throughout the entire neuron. However, they remain inert until they encounter a tagged synapse. The tag acts as a lock, allowing the synapse to capture these proteins and permanently solidify the connection. By requiring both a tag at the local synapse and the availability of global proteins, the brain creates an elegant filter. This mechanism ensures that only significant information is preserved, keeping our memory network efficient and focused. For decades, astrocytes were seen as mere structural glue. However, research as of 2026 has confirmed that these star-shaped cells are actually active, vital architects of our memory. Astrocytes reach out to synapses, forming what we call a tripartite synapse. By surrounding these junctions, they regulate the chemical environment, ensuring that signals crucial for memory are balanced and maintained over the long term. Evidence now shows that without this astrocytic regulation, memory traces quickly degrade. Astrocytes are not just supporting the brain, they are active custodians of our personal histories. Memory is no longer viewed as a static archive of the past. Modern research demonstrates that the brain uses memory to perform what we call in vivo replay mapping. By reactivating past experience patterns during rest or quiet focus, the hippocampus pre-plays scenarios. This simulation engine allows the brain to anticipate future outcomes before they happen. This predictive capacity is essential for cognitive flexibility, turning memory into a survival tool that navigates uncertainty with remarkable precision. Forgetting is often misunderstood as a failure of the brain's filing system. In reality, it is a highly active, essential biological function. Through a process called synaptic downscaling, the brain actively weakens connections associated with outdated or irrelevant information. This prevents cognitive overload. By pruning these weak pathways, the brain preserves metabolic energy and reduces interference. This allows for better focus on new information and keeps our cognitive architecture flexible for future learning. Memory consolidation is not a passive process. During 90-minute NREM sleep cycles, the brain undergoes active synaptic restructuring. By stabilizing daily experiences, this biological mechanism provides a 40 to 60% improvement in long-term retention compared to those who skip sleep. Prioritizing these cycles transforms temporary memories into permanent cortical storage, making sleep the most critical window for academic and skill mastery. Targeted memory reactivation, or TMR, is a clinical tool that leverages NREM sleep to selectively reinforce specific neural ensembles. During sleep, the brain enters a state of slow-wave oscillations. By playing specific audio cues previously associated with a learning task, we can precisely target the memory trace. These cues trigger sharp-wave ripples in the hippocampus, which act as a signal for the cortex to replay the information, effectively consolidating the memory trace. Recent trials have demonstrated significant success in language acquisition, proving TMR is a viable clinical method for stabilizing memory consolidation. Memory retrieval is not the playback of a static file, but a dynamic, reconstructive performance. Every time we access a memory, the neural network associated with it enters a labile or malleable state. In this temporary state, the memory trace can be updated with new information or strengthened by internal associations. However, this plasticity comes with a trade-off. Because we reconstruct the memory from scratch, it can be slightly altered during the process. This is why our memories are inherently predictive tools rather than perfect historical records. Neuroscientists are currently racing to build a complete connectomic map of the human brain. This would be a comprehensive blueprint of every synaptic connection. By mapping these trillions of connections, we are essentially digitizing the structure of human thought. We are attempting to find the physical basis of our memories and personalities. If we succeed in decoding this blueprint, we would gain unprecedented insight into how the self is stored, shifting our understanding of identity from the abstract to the biological. The convergence of neuroscience and artificial intelligence has reached a milestone with neural representational alignment. This methodology allows scientists to directly compare the internal models formed by neural networks with those observed in the human cortex. We observe that both systems organize complex data into specific geometric clusters. When the brain perceives categories like faces or objects, it creates stable patterns of neural activity. Modern AI models, trained on vast data sets, develop remarkably similar internal geometries, suggesting that there may be universal mathematical laws for how intelligent systems encode information. Representational similarity analysis measures how these distances between categories align across systems. Finding these alignments confirms that AI models are beginning to mirror the structural logic of the human mind. This convergence helps us understand that intelligence, whether biological or synthetic, relies on efficient ways to map the external world into internal representations. Advanced neuroprosthetics are now utilizing hippocampal bypasses to assist in memory restoration. When the hippocampus, the brain's primary fast learning center, is impaired by neurodegenerative disease, new memory formation ceases. Brain-computer interfaces, or BCIs, function as a bypass. By embedding electrodes into the entorhinal cortex, the system injects patterned electrical stimulation that mimics natural hippocampal output. This direct intervention effectively bridges the signal gap, providing the entorhinal cortex with the data needed to store new memory traces, essentially rewiring the patient's ability to encode experience. Critical periods are temporal windows of peak brain plasticity, where experiences physically reshape neural architecture at high speeds. Recent neurointervention research suggests we can reopen these windows in adults. By applying targeted sensory or pharmacological triggers, we restore the brain's ability to undergo high-speed structural change. This brings profound ethical questions. Reopening plasticity enables memory enhancement, but it risks neurological instability. Balancing therapeutic restoration with human cognitive autonomy remains a core debate for 2026 neuroethics. Recent research into engram transfer represents a landmark achievement in neuroscience. Researchers have successfully transplanted contextual memory engrams between genetically identical mice. By identifying the specific ensemble of neurons firing during a learning event, scientists use optogenetic priming to tag these cells. This allows the memory to be isolated as a physical circuit. The transfer process involves stimulating the corresponding neural population in the recipient. This demonstrates that memory is fundamentally a pattern of circuit activation rather than a static filing system. Critically, we cannot yet download complex life experiences. However, transferring contextual associations confirms that memories are reconstructive performances, not permanent video recordings. Memory begins with encoding. Here, specific neural ensembles within the hippocampus fire together to form an engram, the physical trace of a new experience. Next is consolidation. This offline process, occurring largely during sleep, migrates information from the hippocampus to the neocortex, where memories are permanently stored. Finally, retrieval is not static. It is a reconstructive performance. When we remember, we reactivate the original engram, rendering the memory temporarily malleable, allowing it to be strengthened or rewired. In summary, we encode via engrams, consolidate via cortical transfer, and retrieve via active reconstruction. This dynamic cycle ensures our cognitive flexibility. Optogenetics has revolutionized how we map the brain. By genetically introducing light-sensitive proteins, known as opsins, into specific neurons, researchers can treat these cells like a biological remote control. When we deliver a precise pulse of light, the opsins change shape, opening ion channels. This forces the neuron to fire, allowing us to observe memory engrams in real time as they form or reactivate. This technology enables researchers to map entire neural circuits with unprecedented spatial precision. We no longer rely on static snapshots. We capture the dynamic electrical performance of memory as it happens within the living brain. A persistent myth in popular culture is that memory functions like a high-fidelity video camera, capturing objective reality perfectly for future retrieval. However, biological evidence from 2026 confirms this is false. Memory is a fallible, constructive interpretation of events. When we recall an experience, we reactivate the original neural engram, but we simultaneously integrate current context and emotions. We are essentially reperforming the experience, not playing back a saved file. Recognizing that memory is constructive is vital. This malleability is a design feature that provides cognitive flexibility, but it comes at a cost. Memories are susceptible to distortion and misinformation every time they are retrieved. To optimize memory, we must move beyond cramming. Spaced repetition leverages the brain's natural consolidation process. By spacing out your review sessions, you allow the hippocampus to successfully integrate information into the long-term storage of the neocortex. Equally vital is the architecture of your sleep. Research indicates that 90-minute sleep cycles are essential for memory locking. During NREM sleep, the brain generates sharp wave ripples, which act as a high-speed replay mechanism, refiring the neurons associated with your learning to solidify the engram. By spacing your sessions and protecting your sleep, you are not just studying, you are biologically managing the reconstruction of your own knowledge. Treat your memory as a performance that requires rest to be perfectly rehearsed. We are entering a transformative era in neuroscience. We are moving from simply observing how memory works to actively assisting it. The future of memory science lies in precision neuroengineering. With emerging brain computer interfaces, we are developing hippocampal bypasses designed to restore cognitive function in those suffering from neurodegenerative disease. This marks our departure from the biological dark ages, leading toward a future where memory is not just a delicate biological process, but a repairable and resilient human asset.